Severe liver disease is often framed as an inherited life sentence, a pre-programmed destiny written into your DNA. Even as we watch the healthy architecture of the liver undergo necrotic cellular degradation, we are seeing the final result of a process that is more adjustable than we once thought. The science of epigenetics shows that our genes are not fixed. They possess binary on and off switches, and environmental inputs actively dictate whether our protective genes are silenced or disease-causing ones are activated. Take hemochromatosis, for example. Due to a specific genetic mutation, the body absorbs massive toxic amounts of iron, which is deposited directly into the liver and brain. When this iron remains unbound and free, it acts as a powerful fertilizer for cancer, triggering intense free radical damage and stimulating rapid, dangerous cell reproduction. We see a similar mechanical failure in Wilson's disease, where a genetic inability to process copper leads to rapid hepatic cirrhosis and neurological problems. The destruction seen in Wilson's and hemochromatosis proves that the liver has a specific, fatal vulnerability to heavy metal accumulation. For everyone else, these same biological pathways are managed by environmental inputs, toxins that act as the toggle for the epigenetic switches governing our long-term hepatic health. The most pervasive threat to these epigenetic switches happens daily in the kitchen, through the inadvertent consumption of a specific chemical complex. This threat occurs when fluoride, commonly added to municipal drinking water, interacts with aluminum leached from standard worn cookware. These two common elements bind to form the fluoroaluminum complex. This is a highly toxic compound that directly damages hepatocytes and generates radiating free radicals throughout the brain. Black tea is a primary source of this exposure, as the plant is a natural bioaccumulator, pulling high levels of fluoride and aluminum from the soil. A simple clinical pivot mitigates this hazard, substituting black tea with younger, less developed white tea. White tea is harvested earlier in the plant's life cycle, meaning it has significantly lower levels of toxic metals, while containing much higher concentrations of protective EGCG antioxidants. Restoring liver function requires actively blocking the stealth bioaccumulation of these epigenetic neurotoxins. Simply washing your produce in the sink is ineffective against the next class of agricultural chemicals. Many modern pesticides, like glyphosate, are systemic. They are absorbed directly into the intracellular structure of the plant and cannot be scrubbed off. Genetically modified Roundup-ready soybeans are specifically engineered to survive heavy saturation with these chemicals. Soybeans also naturally contain an estrogenic hormone-mimicking compound called genistein. When you combine genistein with the extreme endocrine-disrupting power of glyphosate, it creates a compounded hormone-disrupting cocktail. This biochemical combination contaminates consumers even at doses 800 times lower than standard EPA safety limits. Consuming standard commercial soy products delivers a systemic intracellular payload of epigenetic disruptors straight to the liver's hormone receptors. When liver degradation advances to hepatocellular carcinoma, we have to look at why conventional treatments often fail to stop the disease. Tumors are driven by cancer stem cells. You can think of the stem cell as a bubble blower, while the bubbles themselves represent the bulk of the tumor. Conventional chemotherapy and radiation focus on popping the bubbles, killing the bulk mass of daughter cells. While the daughter cells die off, the originating cancer stem cell, the bubble blower, remains untouched and active. This surviving stem cell rapidly generates a new tumor mass, often with a mutation that makes it completely resistant to multiple drugs. Targeted botanical interventions like nanocurcumin, nanoquercetin, and pterostilbene are required to solve this. These compounds selectively bypass the bulk mass to initiate apoptosis directly in the cancer stem cells, while protecting all healthy adjacent tissue. True oncological reversal requires eradicating the origin of the cellular lineage, not merely managing the expanding biological mass. Beyond its structural origin, the liver tumor has a specific metabolic engine, an absolute reliance on massive quantities of the amino acid glutamine. Cancer cells rapidly convert incoming glutamine into glutamate, which accelerates growth and makes the tumor highly invasive. The natural plant extract berberine acts as a specific mechanical intervention against this precise metabolic pathway. Berberine acts as a physical barricade, actively locking onto and blocking the cellular transporter that allows glutamine to enter the cell. Hepatocellular carcinoma cannot be fought with toxicity alone. It must be actively starved by exploiting its rigid metabolic dependencies. In end-stage liver disease, transplantation is sometimes the only option, but it is a high-stakes transition. The primary causes of transplant failure are severe postoperative infection and organ rejection driven by intense cellular inflammation. A clinical study compared patient outcomes on a standard hospital diet against those receiving an omega-3 DHA emulsion. The results showed a dramatically lower one-year death rate for the DHA group. DHA naturally inhibits the inflammatory cytokines responsible for triggering organ rejection. Patients receiving the omega-3s also had significantly shorter hospital stays and fewer postoperative infections. This proves that highly targeted lipid nutrition can successfully suppress systemic immune rejection, where standard pharmacology often fails. The daily foundation for liver healing is a highly modified anti-inflammatory Mediterranean or DASH diet. But the success of this protocol relies strictly on absolute, non-negotiable dietary exclusions. First, zero dairy. Milk and cheese are high in calcium and glutamate, a combination proven to stimulate the growth of cancers, particularly in the prostate and liver. The second absolute exclusion is all whole grains, challenging standard nutritional advice. Whole grains are extremely high in gluten, inflammatory lectins, and excitotoxic glutamate, all of which damage the intestinal lining and burden the liver. Finally, avoid all fruit juices, allowing only moderate whole fruit consumption. Removing the plant's fiber delivers a massive, concentrated liquid dose of fructose directly to the liver, driving rapid fat accumulation. True liver restoration demands deliberately blocking epigenetic toxins, starving malignant metabolism, and enforcing uncompromising systemic dietary exclusions.